Hello, welcome everyone. Very good evening, all of you. Very good evening, all of you. Welcome everyone. All my IP student boards are going on. So stay right now. Connect to this YouTube class. This is specially for IP student and those who are preparing for Neet. Also, they can attend this class for Neet purpose also. Welcome everyone. All my So can I have some quick comments, please? Can I have some quick comments? If you can hear my voice and if you can see the screen. Very good evening, all of you. This is Vaishali Kamdar from Spark Academy. I welcome you, all of you, in channel of Spark Academy. And today we are going to discuss about photosynthesis. So if you will stay with me next 60 minutes, you will be clearing all your doubts related photosynthesis. So we will be discussing PYQs also with solutions. So before starting the topic, let me tell you, you can follow us on Spark Academy on online.com, Instagram, Telegram, Facebook. Spark Academy is there, so you can follow any of the things. So you will be having notification about which all classes will be going on and what all courses we are providing right now. So this is a complete theme. This is a complete theme of Spark Academy. So for MSET, JEE, Neet, we are conducting on daily basis. We are conducting live classes on YouTube that is free. Okay, so free material in app. If you download the Spark Academy app, you will be having free access to all these things. So daily live classes, in session doubt clarification, free materials in our app, tips and tricks we will provide, and PYQs we'll be discussing. And if you are still worried about your NEET exam, so crash course, we are starting a new batch soon. Short term regular already going on. You can join the regular batch for 2023 also. You can join online or classroom coaching, whatever is convenient to you. So this is our well-experienced staff. You can check the timetable or schedule, which is for the live classes on YouTube. So these this is the schedule. You can have a screenshot of it. You can take the screenshot. Come on, screenshot. Take the screenshot. Otherwise, you can do one thing. Subscribe to Spark Academy channel and press the bell icon, so you will not miss out any of the lectures. This is me, Vaishali Kamdar, and we will be discussing photosynthesis today. And if you want to enroll for any of the courses, you can apply my code, that is VAI10, to get a huge discount on all our courses. So my code is, remember the code guys, if you use this coupon code, that is VAI1010, VAI10, then you will be having a great discount on all our courses. So for Neet also, this is very important chapter from plant physiology. You all know, very important, plant and human physiology. For Neet, it is very important. Weightage wise, if we can see the plant physiology, 9 to 10 questions you can expect from the plant physiology itself. So that means around 40 marks. And from the plant physiology, we will be discussing photosynthesis in higher plants today. So photosynthesis, it gives you around 2 questions. So 1 to 2 questions you can expect from all the chapters of the plant physiology. 5 chapters are there. So 1 or 2 questions you can expect from any of this. Photosynthesis, mostly 2 questions are asked from photosynthesis. So let's start. Good evening, Bhavna. So let's start. Photosynthesis. What is photosynthesis? See, simply autotrophs. Simply autotrophs. They will use inorganic. So this is an anabolic process. So they'll use inorganic substances, simple inorganic substances like carbon dioxide and water to make organic compound. This is final product. This will be your product. Okay. Organic compounds with the help of sunlight as a source of energy. So whenever you are cooking anything, you require some or other type of energy, either electric energy or directly fire you will be using. So mainly you require that is heat. Okay, here also we will be using energy. Which source of energy will be using? Plants will be using sunlight as a source of energy. And what they'll be cooking? They'll be cooking glucose after that, sugar. So that is organic compound. And what will be the ingredient for their for cooking sugar? So that is carbon dioxide and water. So importance of photosynthesis includes primary source of food. If plants will not produce this food, what we will have? What we will eat? We all heterotrophs depend on the producers to produce the food. And one more thing, plants help during the photosynthesis. They will also release oxygen in the atmosphere. So this is one of the byproduct. So one thing, you are getting food from the photosynthesis. We all are getting food by the help of photosynthesis, and in turn along with it we will be getting oxygen also. So two main important benefits of photosynthesis. So early experiments, if you can't see the yellow color here, wait. So we will be discussing experiments. So Joseph Priestley in 1770 performed a series of experiments with bell jar, candle and mouse. So these were their experiment. What he has done? He kept a candle. Inside the bell jar. Candle will extinguish obviously. Okay. Then he kept mouse inside the bell jar. Simply mouse alone. Then by suffocation mouse died. Then he kept both together. Then both candle extinguished and mouse died. You can see the first bell jar. Then what he has done? He kept mint plant along with the candle and mouse. Then candle keep on igniting. There will be no extinguishing of the candle occur, and mouse also remain alive. So that means he concluded that there is something in the air which is used by the candle and mouse. So that means they are damaging the air. And which is restoring by the plant. The air which is restored by the plant. So that is what Priestley said. What he said? Mouse would soon suffocate. So he concluded that the burning candle and an animal that breathe the air, they somehow damages the air. They damage the air. And the mint plant in the same bell jar, it restored the same. So plant restore to the air whatever breathing animals and burning candles remove. Now we are knowing what is removed by the plant. What is removed by burning candle and mouse? Come on, Bhavna. What is removed by burning candle and mouse from air, which is restored by the plant? Can you tell me? Anyone? Those who have joined? Come on, guys. If you want me to call out your name, you can answer. You can chat in form of answer in form of chat. Yes, Bhavna. What is removed by air? What is removed from the air which is restored by the plant? Correct. So what is removed? It is oxygen. It is oxygen which is removed. Okay. So that oxygen is getting restored by plant. How plants are restoring oxygen? With the help of photosynthesis. Bell jar, why he has taken bell jar, not any cardboard box? Because bell jar, it is transparent, it is made up of glass, and that's why sunlight, plants can use the sunlight for photosynthesis. Next experiment was done by Ingenhousz. So Jan Ingenhousz in 1730 and 1799 showed that sunlight is essential to the plant process that somehow purifies the air, followed by the burning candle or breathing animals. Okay, so again what I have told you, why he has not used cardboard box. So what engineer was saying, what he is concluding, that for this restoration, sunlight is essential. So use of sunlight for photosynthesis has been proved by Ingenhousz. Then he performed experiment with the aquatic plant and showed that in bright sunlight, some bubbles were formed around the green parts, while in dark they did not. So when sunlight is there, bubbles were formed, and these bubbles were of which gas? Oxygen itself. In dark, it won't. So later he identified those bubbles to be oxygen. Hence he showed that it is only green part of the plant that could release oxygen. Okay, that means green part, what he has concluded, one thing, that is sunlight. About sunlight is necessary for photosynthesis, and second thing, that is only green part of the plant can release oxygen. Green color responsible for photosynthesis. Next, Julius von Sack proved that plant produces glucose while growing and stores them as a starch. So, first product of the photosynthesis is glucose, and it is stored in form of starch. He explained that plants have green substance which is present within a cell in a specialized body. Now we are knowing what is the specialized body in plants? Chloroplast. So this chloroplast, they have some green substance. We all know about that green substance, green pigment, chlorophyll. So he found that green parts in the plant is where glucose is made, and that the glucose is usually stored in form of starch. Then Julius von Sack proved that plant produce glucose while growing and store them in form of starch. He observed that the bacteria accumulate mainly in the region of the blue and red light. Bacteria, they were accumulated so this has been proved by Engelmann. In 1843 to 1909. So he used the splitting of the light into the spectral component, and illuminated a green algae that is cladophora. So he has used algae cladophora and used the spectrum, and then he has concluded that the bacteria accumulated mainly in the region of the blue and red light of the split spectrum, and it explained the absorption of the spectra of the chlorophyll A and B. So this is how he explained that absorption spectra of the chlorophyll pigment is in blue and red region. So key feature of the photosynthesis was first explained in 19th century, that plant could use the light energy to make the carbohydrate from water and carbon dioxide. So empirical equation representing the total process of the photosynthesis is CO2 plus H2O gives you CH2O plus O2. So what is the CH2O? It is actually a carbohydrate, represented a carbohydrate. So then when Neil, he did the experiment with the purple and green bacteria, demonstrated the photosynthesis is a light dependent process. So he explained hydrogen from the H2O. Hydrogen from the H2O reduces to the carbon dioxide, carbon dioxide to the carbohydrate. See what is actually reducing? Who is reducing? This hydrogen of water is reducing carbon dioxide to what? Carbohydrates. So we concluded that oxygen comes from water, it is not from carbon dioxide. So we will discuss how oxygen comes from water. First step of photosynthesis is photolysis of water, and in that case oxygen is released. So it is actually from water, not from CO2. Then site of photosynthesis, very important for IPE exam also. They can ask you different sites of different things, which is a site for ATP production, biosynthetic phase occurs in which part of the chloroplast, like that. So important for short questions. So photosynthesis takes place only in green part of the plant, mostly in the leaves. Within a leaf, photosynthesis occurs in mesophilic cells, which contains the chloroplast. We all know about leaf structure. In anatomy, we have discussed if it is dorsiventral leaf, number of the chloroplast or number of stomata will be higher in lower surface of the leaf, and we all know about the dorsiventral leaf structure that there are mesophilic cells are there, and two types of mesophilic cells. These type of columnar type mesophilic cell and like this some cell, which contains, we are making dot like structure like this. What is this dots? These dots are chloroplast in it. Okay. Palisade mesophilic cell and spongiform mesophilic cell. In case of isobilateral leaf also, there is only one type of mesophilic cell. So photosynthesis occurs in mesophilic cell, which contains chloroplast. Chloroplasts are actual site of photosynthesis. Now what is the structure of chloroplast? We'll first look into the structure. Look here. So chloroplast, you can see the membranous structure here. So these all are membranous structure. The single, see, if you think about the coins, bunch of a coin stacked one over the other, then this complete bunch is known as granum. This is called granum. And one part of each that is, I mean, one coin you can consider, it is known as thylakoid. Okay, these all are membranous structure. So all granum together they form grana. And between two grana you can see this adjoining thing that is known as grana, that is called stromal lamina. That is called stromal lamella. And this matrix part of chloroplast. Matrix part of chloroplast is known as stroma. I hope it's clear, guys. Just double membrane bound organelle. You can see the outer layer and inner membrane, inner membrane, then membranous structure. Membranous structure is formed by grana, and matrix type structure or semi-fluid type structure, it is formed by stroma. So chloroplast, thylakoid in the chloroplast contain most of the pigment required for the capturing solar energy. So pigments are present mainly in thylakoid. That means a single coin in that this pigment's chlorophyll is present. So membrane system grana is responsible for trapping the light energy. Light energy is trapped by the pigment actually. Okay, pigment is present in grana membrane system. So that's why it is important for trapping the light energy and for the synthesis of ATP and NADPH. So in first phase of photosynthesis, so it is responsible for, in first phase it is photolysis of water. We will discuss that. So there are two reactions, light reaction and dark reaction, or we can say biosynthetic phase. So this biosynthetic phase or dark reaction, it is carried out in stroma where actually carbon dioxide is reduced to carbohydrates. So pigments involved in photosynthesis. Chlorophyll A, bright or blue-green in the chromatograph. So if you want to separate the pigment, if you want to study the pigments, you can simply extract out the pigment and you can carry out paper chromatography of pigments. Okay, so you will get different color bands in paper chromatography. You will require mobile phase and stationary phase. We shall discuss this type of chromatographic technique later on. So simply by chromatography, pigments are separated, or you can have different type of pigment extracted and afterwards separation in form of chromatograph. So this is also one of the technique, important questions, short answer questions for IPE as well as Neet. How you can get different bands or how you can separate pigments from the leaf. Extraction, after that you can carry out chromatography. So major pigment acts as a reaction center involved in the trapping and converting light into the chemical energy. So chlorophyll B, it's yellow-green, xanthophyll is yellow, carotenoid is yellow to yellow-orange. So in blue or red region, spectrum shows the higher rate of photosynthesis. So in NCRT or in your Telugu Academy book, so in that there is one graph given for this rate of absorption, and this I'll tell you which graph. Yeah, absorption of light by chloroplast pigment, and which is correlated with the rate of photosynthesis. If you think where actually photosynthesis occurs in a higher amount of photosynthesis occurs, then it is also in red and blue region, and the amount of absorption maxima of chlorophyll A is also in the same region. So that's why we can conclude that actually blue and red region of the spectrum shows the higher rate of photosynthesis. Then what is light reaction? First point, important point of photosynthesis, light reaction. So process which takes place in presence of light only. Without light, this cannot happen. So light reaction or photochemical, it is also known as photochemical phase. So splitting of water, evolution of oxygen, and formation of high energy compound like ATP and NADPH. So it involves several complex, and the pigments of the complex are organized into two discrete photochemical light harvesting complex. So there are two discrete light harvesting complex, LH3, LHC, PS1 and PS2. This 1 and 2 name has been given not according to their role in the photosynthesis but according to their discovery. First discovered photosystem is photosystem 1. That's why we have given its number 1. So light harvesting complexes are made up of hundreds of pigment molecules bound to a protein within the photosystem 1 and 2. So hundreds of pigment molecules forming what is how light harvesting complex LHC are, or what how it is made up of. It is made up of hundreds of pigment molecules bounded to a protein. Bounded to a protein, that means protein plus hundreds of pigment molecule. Note it down guys, forms the LHC. LHC, that is light harvesting complex. Important points. See guys, if you are making your notes along with me, you can quickly revise the chapter right now. And if you are writing all these things in your chat box also, or you try to write along with me, whatever I am saying, it is important. Whatever I am underlining, you should mention it in your quick notes book. Okay, as I have told you, एनसीईआरटी को तो हम सजा ही देते हैं आजू बाजू लिख के, but still you need to prepare one quick notes book which you will be referring before 2 hours of your exam. Okay, important points of each and every chapter. So you can make notes right now along with me. So this is what pigment molecules, but reaction center is only one. See, it is just like a team leader. Whatever we are preparing, we should give to the team leader, or we should give it to the leader. So leader is only one molecule. It is forming the reaction center. All other molecules, all other pigment molecules, they trap that light, but they will be transferring that energy to the reaction center itself. I hope you are getting this thing. Each photosystem has all the pigments bound to a protein except one molecule. Remember this one molecule? ये कौन है? This is the leader, team leader. Except one molecule of chlorophyll A. One molecule of chlorophyll A, forming the light harvesting system antennae. And pigment absorbs all other pigments absorb the different wavelength of light for the efficient photosynthesis. And reaction center is different in both the photosystem. In photosystem 1, chlorophyll A has absorption maxima, absorption peak at 700 nanometer, and PS2, it is 680 nanometer. Now photosynthesis process, it includes two phases as we have discussed, light and dark, photochemical and biosynthetic phase. So first, electron transport system. So in photosystem center, chlorophyll A absorbs 680 nanometer wavelength of light. Okay, causing the electrons to become excited. What happens? 680 वाला कौन है? PS2. So that is PS2 is absorbing this light. Okay, and this is how electrons become excited. Okay, when they are excited, they'll be moving in the farther electronical orbits. They will be moving in the farther orbits through the nucleus. So they release two electrons from the atomic nucleus. These electrons are accepted by excited electrons are accepted by primary electron acceptor ferredoxine. First, it is ferredoxine. Then electron from the ferredoxine pass to the electron transport system, that is ETS, consisting all the cytochromes and all. We will be discussing this in next class, that is respiration. In that we will discuss detailed electron transport system. So the electron moved in the downhill in terms of the redox potential. See, first it will move uphill. First it will move uphill when it is getting excited. Then again it will move downhill. Then again finally electron reach to the PS system 1. PS1. Okay. So this is how what it happened. Look here. Again I will explain. Light is absorbed by LHC, that is PS2 first here. Okay. Then electrons get excited. These electrons are accepted by primary electron acceptor, mostly ferredoxine. Then it will transfer it to ETS. During this process, ATP is released and then electrons are again transported to PS1. Photosystem 1 at 700 nanometer, it can absorb the light. Okay, again electrons present here, they get excited, which will be accepted by other electron acceptor. And finally, it will go again downhill and it will be transferred to the NADP reductase. NADP reductase, and there will be formation of NADPH by then. Okay, during this process, okay, these electrons comes from where? Photolysis of water. From photolysis of, this is important step. From photolysis of water, 2 electrons will come here. Okay, and oxygen also getting released. Correct? So as it is uphill, first you are going up, then you are going down, then again you are going up, then again down. So like this, it is somewhat like, if you see slantly, okay, then you can see this is theta z. Okay, so that's why they have actually, from the front we can see it is L shaped, but I don't know why Z pasand aaya hoga unko. So that's why they have given the scheme, name of the scheme as Z. See, it is like this. So it is zigzag type thing. So that's why they have given name Z scheme. And this is the same thing in non-cyclic photophosphorylation. This is also known as non-cyclic photophosphorylation. So electrons are not used up as they pass through the electron transport chain, but they are passed onto the pigment of the PS1. Now electrons in the PS1 get excited and transferred to the another acceptor with the greater redox potential, that means uphill, and then PS1 is filled up by the electron from the PS2. Electrons from the PS1 pass downhill to reduce NADP to NADPH plus H plus. So during this process we can observe this Z type of shape, that's why we call it as Z scheme. Splitting of water. Electron required for the electron transport system is fulfilled by the splitting of the water photolysis. Photons are accepted and water is split. Okay, water is split into H plus and O nascent oxygen and electrons in presence of the light and manganese ion and chlorine ion. Remember names of the ions here, important point guys. Note it down. For photosynthesis, which inorganic substances or which inorganic ions are required? Manganese as well as chlorine. So this will give you 4H plus. In 2H2O you will get 4H plus and O2 and 4 electrons. Then photophosphorylation, there are two types, cyclic and non-cyclic. Non-cyclic we have discussed in this Z scheme. What is cyclic one? So only PS1 works if only PS1 works, electrons circulate within the photosystem. PS2 is not involved here. So it happens when the stroma laminae, possible location of the photophosphorylation is stroma laminae, because in this region PS2 and NADP reductase enzymes are absent. So only ATP will be produced, no NADPH will be produced in this reaction. Okay, so hence product is only ATP. Again mention this point guys. Cyclic. All my students, please note it down right now. In cyclic, what are the products you will get in cyclic phosphorylation? Only one ATP. And in non-cyclic, you are getting NADPH plus H plus also along with ATP. This NADPH is high energy molecule, which will be again giving you three ATPs afterwards. We will discuss how it will give you 3 ATPs in respiration chapter. Next, chemo-osmotic hypothesis. So, chemo-osmotic hypothesis has been put forward to explain the mechanism of ATP synthesis, which is linked to development of the proton gradient across the membrane of the thylakoid. See how ATP is produced. We all know that Shakti kahan se aati hai? From where energy is, from where all the organisms they are getting all energy by the reaction of respiration. So this is the synthesis process. We are making the food, and along with the making the food, we'll be using energy and we are getting energy also. ATP is also synthesized. So what is the reason? Okay, where actually the reason lies, how ATP is synthesized during photosynthesis. Actually, ATP should be synthesized in mitochondria only during respiration, right? So this is the hypothesis which explains how ATP is synthesized, how ATP synthesis occurs here. One second. So as it is, see, as you all know that during photolysis, photolysis, hydrogen ions are also generated. So this proton gradient across the membrane of the thylakoid, this is in chloroplast ATP is generated. Okay, not in mitochondria. So here the proton accumulation takes place towards the inside of the membrane, that is in the lumen. Thylakoid, what is thylakoid? A one coin, simply a coin type structure. Okay, if you think about the complete chloroplast, then this is grana, this is granum actually. In that one of that coin, in that actually inside it, it is known as lumen. So inner part of the thylakoid actually there. Okay, so that is called lumen. In the lumen, accumulation of the proton is there. So proton that are produced by the splitting of water are accumulated in lumen. And as electrons move through the photosystem, protons are transported across the membrane. These protons are transported outside the membrane. Okay, so NADP reductase enzyme is located on the stroma side of the membrane along with electrons from the acceptor. It removes H plus from the stroma during the reduction of NADP H plus H plus. So this creates proton gradients across the thylakoid membrane as well as measurable decrease in the pH in the lumen. Remember, when H plus ions are removed from the lumen of the thylakoid, at that time there will be decrease in the pH of lumen. And ATP has the channel that allows the diffusion of proton back to the stroma across the membrane. This releases energy to activate ATP as enzyme which catalyze formation of ATP. Finally, this H plus ions are given to ATP as enzyme which will form ATP. So chemoosmosis requires a membrane, a proton pump, a proton gradient and ATPase. So these are the requirements for chemoosmotic hypothesis. Remember guys, proton pump, membrane, proton gradient, that is H plus and ATPase. So energy is used to pump the proton across the membrane. You all know that ions cannot be transported just like anything. Simply bhej diya like that. It could not transport it. For that we require some ion channels. Okay, pumps like sodium potassium pumps. So these pumps are required when you want to transport ions or protons across the membrane. So to create the gradient or a high concentration of protons within the thylakoid membrane. So this is what a chemoosmotic hypothesis is showing. This pink color diagram, what you can observe in your screen. So this is actually thylakoid. This is actually one coin, single coin of your granum. So this is thylakoid. You can see that this is thylakoid membrane and inside that this is called lumen where actually these protons are accumulated and these protons are transported across the membrane where it is coupled with the ATP synthase enzyme which catalyzes this ATP formation. This is the reason for formation of ATP during photosynthesis in chloroplast. Not in mitochondria. Mitochondria to powerhouse hai. But right now during photosynthesis also one ATP is produced. How? That we have discussed. Now how ATP and NADPH is used. See, you require energy. Again, you require energy to synthesize carbohydrate. है ना? So light energy we are transferring, plants are transferring their light energy in form of chemical energy. They will first synthesize ATP and NADPH, high energy molecules. Then these high energy molecules they'll be using for biosynthetic phase where actually carbon dioxide is reduced in form of glucose. So in this ATP and NADPH are used here. So Melvin Kelvin, after the World War II, name of a scientist, Melvin Kelvin, after World War II, worked with the radioactive carbon-14 and studied algal photosynthesis, which led to the discovery of first CO2 fixation. This carbon dioxide is fixed in form of C6 C6H12O6, that is glucose. So product was 3 carbon organic compound. First CO2 is first product of this carbon dioxide fixation is 3 carbon organic acid, that is 3 PGA. So this CO2 is fixed by first step, it is 3 PGA. Okay, then finally it is converted to glucose. So he explained a complete biosynthetic pathway and named that Calvin cycle. So further work of the scientists led to the discovery of another set of plant in which first stable product is 4 carbon compound. Normally it is 3 carbon compound. That's why it is known as, actually it is known as C3 pathway. But in some other set of plants like sugarcane, so first product of CO2 fixation is oxaloacetic acid, which is 4 carbon compound. So it is known as C4 pathway. So photosynthesis, in the process of photosynthesis start with the CO2 is accepted by the primary acceptor. So always remember primary acceptor is 5 carbon ketone sugar, that is ribulose bisphosphate. So next step is in the Calvin cycle, it is carboxylation, reduction and regeneration. We'll discuss this thing. Look here. So first CO2, look here in the atmosphere, whatever CO2 is there, it is accepted by ribulose bisphosphate, 5 carbon compound. So 5 carbon plus 1 carbon gives you 6 carbon compound. This is called carboxylation. Okay, 6 carbon compound is actually in form of 2 molecules of 3 PGA. That is, first it is fixed in form of 3 carbon compound. Okay, so that is PGA, phosphoglycerate. Then it is reduced. It is reduced by the use of ATP plus NADPH to triose phosphate and finally sucrose. Okay, then after that again only 1 carbon dioxide is here. This after 6 cycles 1 molecule of glucose is fixed, and after that regeneration of RUBP occur, and at that time 1 ATP is used here. So first step of carboxylation, Calvin cycle, carboxylation is fixation of CO2 into stable organic intermediate. So it is 3 PGA, then reduction, series of reduction that lead to the formation of glucose molecule, involves utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction of CO2 molecule. And the fixation of the 6 molecule of CO2, see, look here, 6 turns of the cycle required, 6 turns of the cycle required here for fixation of 1 glucose molecule. Then regeneration of the CO2 acceptor molecule, RUBP, is crucial if the cycle is continued uninterrupted. So regeneration steps require 1 ATP for phosphorylation. Remember, there 2 ATPs were required here, 1 ATP again required. So for every CO2 molecule entering the Calvin cycle, you require 3 molecules of ATP to NADPH. So this is what 2 ATP requires, and 3 ATPs and 2 NADPH required. Then C4 pathway, here first stable compound which is formed after fixation, first step, that is oxaloacetic acid, which is 4 carbon compound. Okay. So plants that are adapted to the dry tropical region have the C4 pathway. So where there is more heat, they will close their stomata and still carry out photosynthesis. The plants with C4 pathway does follow the Calvin cycle. So Calvin cycle is there. It is not only C4 pathway. But C4 plants differ from the C3 plants by having special type of leaf anatomy, and this special leaf anatomy is known as Krenz anatomy. Look here. So anatomy of the C4 plants differ from the C3 by the having large cells around the vascular bundles, which are called bundle sheath cells. which are called bundle sheath cells. And the leaves, one second guys. So anatomy of the C4 plants differs, and this is known as Krenz anatomy. Krenz, that means something is covered completely. Okay, something, see this type of flower bouquet you might have seen, this circular type of flower bouquet. Okay, so it is like that only. Something is completely covered in the surrounding. What is covered in the surrounding by the bundle sheath cells? This vascular bundle. So that is known as Krenz anatomy. So bundle sheath cells may form several layers around the vascular bundles and have large number of chloroplast thick walls impervious to the gaseous exchange and no intracellular spaces. So pathway followed by the C4 plants are called Hatch and Slack pathway. So the tissue involved in the process of C4 pathway are mesophyll and bundle sheath cell both. So CO2 is accepted by CO2 acceptor, 3 carbon molecule, PEP. Look here, we'll discuss it here. Oh, sorry, that diagram is missing. So actually what happens, I'll tell you. So in the atmosphere, for example, this is your mesophilic cell and this is your bundle sheath cell. There is one channel, plasmodesmata between this. This is mesophyll. And this is bundle sheath. So what happens from here? CO2 is getting inside the mesophilic cell. Okay. Then it is converted. This carbon dioxide is converted to C4 acid first. That is oxaloacetic acid. Then the C4 acid is transferring here to the bundle sheath cell. Okay. There decarboxylation occurs. It undergo decarboxylation. That means one CO2 is released here and C3 acid is formed. And the C3 acid is transported back to the mesophilic cell where it will undergo regeneration and Calvin cycle. Okay, so let us discuss here. So C4 acid oxaloacetic is formed in the mesophilic cell, which then forms the 4 carbon compound like malic acid and aspartic acid in mesophilic cell. This malic acid 4 carbon compound again transported to the bundle sheath cell, and the tissue involved in the process of the C4 pathway are transferred in the bundle sheath cell. The process starts and the bundle sheath cells, these C4 acids are broken down to the release of the CO2, that is decarboxylation, and 3 carbon molecule. So 3 carbon molecule is transported back to mesophilic cell and converted to PEP again, thus completing the cycle. So this is how C4 plant can carry out C3 as well as C4 pathway both. Now factors affecting photosynthesis. The factors which affects the photosynthesis include the availability of the sunlight, temperature, carbon dioxide concentration and water. Yes, Bhavna, you're right. Then Blackman's law of limiting factor. Important point again for IP as well as Neet. If a chemical process is affected by more than one factor, okay, any process is affected by 3-4 factors, say for example, then which is becoming the limiting one? Okay, so that is what a limiting factor law says. So more than one factor, then its rate will be determined by the factor which is nearest to the minimal value. Whatever is at the nearest to the minimal value, It is the factor which directly affects the process if its quantity is changed. First, light. Low intensity light, there is a linear relationship between incident light and carbon fixation. High intensity light, the rate does not show further increase because the factor becomes limiting again. Increase in the incident light beyond a point result in the breakdown of the chlorophyll and decrease the photosynthesis. CO2 concentration also major limiting factor for the photosynthesis. In atmosphere, it is only 0.03 to 0.04%. If you increase concentration up to 0.05, there will be increase in photosynthesis and thereby in carbon fixation. So an increase beyond this level is damaging over the longer period of the time. Then temperature. Dark reactions are controlled by temperature because they are enzymatic. Light reactions are also sensitive to the temperature. So C4 plants have much higher temperature optimum than the C3 plants. They can close their stomata, they can reduce their transpiration rate, and still they can fix more carbon. That's why mass or biomass, you can say, it is increased in terms of C4 plants. The temperature optimum for the particular plants also depend on the natural habitat. Tropical plants have higher temperature optimum than the temperate plants. Yes, Bhavna, what's your doubt? Your question is incomplete, Bhavna. How many what? What is your doubt? Water, the effect of the water is more on the plant rather than directly on the photosynthesis. Water stress is there. Just closing the stomata, you all know that. If guard cells are turgid, then only stomata will remain open. So water stress also lead to the wilting of the leaves, and wilting of the leaves means reduce the surface area, reduce metabolic activity in the leaves. So let me remind you all of you guys, you can download the app, that is SparkAcademy app from Play Store, and you will be having great discounts on all our courses. You can apply the coupon code, that is VAI1010. So that you will be getting all these things. You can purchase all these things from Spark Store. Link is given in the description box. Classroom as well as online coaching is going on right now. Admissions are open for crash course. You can join any of the branch, Narayanguda and Mehdipatnam. For online also, you can use the same coupon code VAI10. That is Vaishali. VAI stands for Vaishali. And on daily basis, on demand, we have this type of live sessions. This is how we are giving assignments and conducting tests. Our method and why only Spark? Because you can feel the difference. We have highly experienced faculty, personalized coaching is there, we have limited batch size, and we can clear all your doubts. Yes, Bhavna, in which process how many ATPs are produced? So first question, now come on, solve your doubts, see whatever ATP is produced in photophosphorylation, this ATP, how that ATP comes from where that ATP comes from, that we have discussed in Chemo osmotic hypothesis, Bhavna. So ATP produced in non-cyclic as well as cyclic is one only. One ATP produced, only one. Very less energy produced though. We need to discuss from where it is coming. So that's why we have discussed. For that reason is chemoosmotic hypothesis. Okay, first question, bundle sheet cells. They are rich in rubisco, they are rich in PEP carboxylase, lack rubisco or lack both rubisco and PEP carboxylase. So answer is A, they are rich in rubisco. Second question, anoxygenic photosynthesis is characteristic of Rhodospirillum, Spirogyra, Chlamydomonas or Alva. N-oxygenic photosynthesis. It occurs in rhodospirillum. Electron donor is inorganic compound during photosynthesis. Organic compound during photosynthesis. Third question. The structure that are formed by the stacking of the organized flattened membranous sacs in the chloroplast are speckled organized membranous sacs. They are called grana, stroma lamini, stroma or cristae. Easy peasy types, grana. They are called grana. Single unit, it is called thylakoid. Fourth question, chromatophores takes part in. Easy peasy again, photosynthesis. Chromatophores play an important role in the photosynthesis. They contain pigments and they are found in blue-green algae. Oxygen involved during the photosynthesis comes from water molecule. Which one of the following pairs of the element is involved in the reaction? I have told you to mark this point. Which elements are involved for this photolysis of water? Manganese, potassium, magnesium, molybdenum, magnesium, chlorine, manganese and chlorine. Correct answer is D, manganese and chlorine. The photosynthesis in the light reaction takes place at photosynthesis, in photosynthesis, light independent reaction, that is dark reaction, takes place in PS1, PS2, stroma matrix, thylakoid lumen. So answer is stroma matrix contains a number of flattened membranous sac called thylakoid or lamellae. Cytochromes are found in outer wall of mitochondria, cristae of mitochondria, lysosome or matrix of mitochondria. They are found in cytochromes. These are located in the inner membrane of the mitochondria and are related to phosphorylation. Next question. So, emergence enhancement effect and the red drop have been instrumental in the discovery of what? Is instrumental in the discovery of B, that is two photosystem operating simultaneously. Next, in chloroplast, the highest number of protons are found in stroma lumen of the thylakoid, intermembrane space or antony. Answer is B, lumen of the thylakoid. We have discussed in chemo osmotic hypothesis. Water-soluble pigments found in the plant cell vacuoles are xanthophylls, chlorophylls, carotenoid, anthocyanins. So they are anthocyanins. Let me remind you all of you that we have a Telegram channel also, so you can click on this link to join us on Telegram. These are your homework questions. Take a screenshot and I'll be waiting for your answer. Yes, Bhavna, I'll be waiting for your answer in comment section afterwards, after completion of the class. You can just take a screenshot and check the correct option. Second question, second homework question. Which one of the following categories of the organism not involve oxygen during photosynthesis? They cannot evolve oxygen. One of the product is not oxygen. Pigment containing membranous extension in some cyanobacteria. We have discussed this during the class. Still I am asking, just waiting for your comments. This is our result. 2021 MSET result. 2021 Neet result. Graph of that, highest score 97%. So do not forget, if you like the video, do not forget to like, share and subscribe. Don't forget to press the bell icon for the notification. Do not forget to press the thumb icon for liking. See, I'll be checking that likes and I know that you are liking my videos. So how you can show that you like the video? Just press the thumb icon also. Thank you so much. Do not forget to like, share, comment for the answers. Thank you so much.